So hi everyone. I would like to thank the organizers for giving me the opportunity to present to you today. So my name is Moran Frenkel-Pinter. I'm a NASA postdoctoral fellow working with Dr. Hudd, Dr. Williams, and Dr. Grover here at Georgia Tech and in the Center for Chemical Evolution. And today I will tell you about some reversible polymerization of prebiotic Depsipeptides. I will define what these are, which allow selection of stable structures. So as we know, and as mentioned, we know from prebiotic model reactions and from meteorite composition that actually all of today's, the building blocks of all of today's biopolymers can be formed from these model periodic reactions, and we can find these in meteorites. But something really important is that in the Center for Chemical Evolution, we explore other proto-backbones for these biopolymers we see today. And we know that in addition to these building blocks that we see in today's biology, we actually had very similar building blocks that could have been incorporated into proto-backbones of these polymers in the prebiotic Earth. So David talked about what were plausible proto-nucleobases, and what I will be talking on today, or be focused on today, is what we think were the proto-peptide backbones on the prebiotic Earth. And today we know these are just chains of amino acids. But we think that we had some incorporation of other building blocks into them. So just in general, I would like to mention in the Center for Chemical Evolution, what is our approach for polymerization of these polymers. And Becky mentioned, and David, dry wet cycles. So just a brief overview. Becky, you said you like the sun. We also like the sun. So as a driving force for polymerization, we know that all of today's biopolymers are a result of condensation reactions, so we lose water. And that's exactly what we want to mimic by these dry wet cycles. So we can think about these, let's say, seasons on the prebiotic Earth. We had some solutions with some monomers. Then Earth we had a dry, hot phase. Water were evaporated. That caused the condensation or polymerization of these monomers. And then we can think about Earth got cooled and we had some water coming, and then we can have some structures and also some hydrolysis or degradation of non-structured polymers. That will allow us some recycling, and we can think about this cycle going on and on, wet-dry cycles, to drive selection of some stable polymers. The outline of the talk today is, so I'm going to start with some challenges with contemporary proteins or peptides in terms of, or in light of chemical evolution. I will talk about what are Depsipeptides as an alternative peptide backbone. And I will also show you some selection of stable structures in a complex mixture. So let's start. When we think about the protopeptides, we know that peptides today are just chains of amino acids, but we also know it's pretty hard to form the amide bond between the amino acids. This is not a thermodynamically favored reaction, and we know it can be formed if we use really high energy molecules, or if we'll apply some really hot temperature, high pressure, we can form that. But then we'll run into another problem, which is once we have this dipeptide, it can go another condensation into a cyclic diketopyrazine, which is really stable. And in a way, it's going to, many times we'll call it a sink. We're going to stop polymerization into further elongated chains. Another problem is that reversibility. So if we want to drive selection in our system, we think that, so we can think about the prebiotic soup as really messy, as having these really, as having a really huge sequence structure space we want to sample. And if we made these peptides, and we cannot break them, so the amide bond, in addition to being hard to make, it's also hard to break. So we can form these peptides, but then we're kind of stuck with what we had. So how can we have selection if we think about really trying out different sequences and give us some stable structures? So we're going to be able to be stuck. We're going to be able to not move forward, and we'll just be maintain with this certain sequence that we had, so we will not have reversibility. So when we search for a protopolypeptide backbone, I mentioned that in addition to the building blocks of today's peptides, the amino acids, we had other very similar molecules. In this case, I'm going to talk about hydroxy acids. So in these molecules, instead of the amine, we have the alcohol or OA, and we know that they result from the same synthesis. So we had hydroxy acid in a really similar abundance to amino acids. And so I will, throughout the lecture, I will just, out of simplicity, amino acids will be labeled as these blue circles, and hydroxy acids will be represented as these red circles. As I mentioned, amino acids, it's really hard to polymerize amino acids into these amide bonds. But what we do know is that if you take hydroxy acid and you just dry them, so you dry water off, it's very easy to form the polymer of hydroxy acid, which is called a polyester, through an ester bond right here. So that's really easy. So what Dr. Krishnamurthy suggested as part of the research in the Center for Chemical Evolution is what will happen if we'll take these both amino and hydroxy acids together. So what happens is we'll actually get copolymers of both amino and hydroxy acids. They are called Depsipeptides. And briefly, I will not go over the mechanism, but I'll just say that once we form the ester bond between hydroxy acids, then the amino acid or the amine can attack and replace, and we get a replacement of the ester with an amide bond. So we're making depsipeptides. In support of a potential role for depsipeptides in origins of life, we know that even today we have depsipeptides in nature. They mostly serve as antibiotics, but they also have different functions. This one, for instance, is called valinomycin. It's a cyclic dodecadepsipeptide, and it functions as a potassium transporter that facilitates the movement of potassium ions through lipid membranes. So as I mentioned, depsipeptides, they're readily formed under model prebiotic reactions, so we can just dry together amino and hydroxy acids, and we can get them. But the problem is that it's not so simple. So if we just take these monomers, we actually get a very complex mixture of depsipeptides. And my research goal focuses on better understanding of these depsipeptides. And if we have this complex mixture, then how can we really study the properties of depsipeptides in terms of polymerization, assembly, disassembly? So a way to make it more simple is really knowing the different stability of the ester versus amide bond. So here we rely on chemistry, and we know that actually the ester bonds, so left to the red circles, the ester bonds are really more susceptible to being hydrolyzed in the wet phase, so when we have water. So if we're going to break all these bonds and maintain the amide bonds, we're going to be left with peptides like this, and we know that this is what happens in the system. So to simplify the system, what I did in my research is synthesizing these peptides, which have just one hydroxy acid followed by an amino acid. And I'll show you one example. So this is glycolic acid alanine. So glycolic acid, it's a red-blue molecule. So we have the hydroxy acid analog of glycine, that's glycolic acid, and alanine. And then if we take this molecule and we just dry it, so we dry water off, we dry and heat it, our expectation is that it will form an ester bond between the two units. And that's exactly what happens. So here you can see an HPLC analysis, separation on a C18 that's based on hydrophobicity. You see absorbance at 210 nanometers. That's going to give us the absorbance of the ester and amide bonds in the system over retention time. So the shorter the retention time, the more hydrophilic the compound is. So as time goes by, we will see the longer species, which are more hydrophobic. You can see over drying time, this is at 65 degrees Celsius, that we start to form these, we start to stitch these units together. And it's really nice because everything that we do really is forming these astro bonds. And what we're making is a new backbone, which is hydroxyamino acid. Then we ask, are we going to get some structures once we polymerize these peptides? So when we start with glycolic acid alanine, these properties are shown in the scanning EM right here, scanning electron microscopy. These samples or these peptides are really oily-like in nature. But once we polymerize or once we dry them, in addition to seeing the polymers, we also start seeing some structures formed, in this case, these fibrils. And we think that what happens is that once we took this glycolic acid alanine and we polymerized it, we think that we're mimicking a really well-known motif in biology, which is the glycine-alanine repeat motif from silk fibrines, in which they form, the glycine-alanine repeat form these nice beta sheet structures. So we think that once we polymerize the peptide, some portion of it can form these fibrils, similar to the glycine-alanine in silk. The next question is, can we go back? So we have the structure formation, we have the polymerization, but I told you reversibility is a really important issue. So can we go back and hydrolyze it and go back to the starting material? And the answer is yes, we can. So if we take the preformed oligomers, so now we have many ester bonds, you can see incubation in water at 65 degrees. So we can actually go from a distribution of oligomers to a shift towards the lower molecular weight species, so actually degradation. And I want to say that the degradation is, again, just of the ester bonds, so left to the red molecule. So we have this unit, in this case, a hydroxyamino acid that is being stitched together when we dry it, and it's being degraded as a unit when we hydrolyze it. So we have reversibility, and the next step was making the system a bit more complicated and see what happens when we have mixtures of these short peptides. And in collaboration with Martin Solano, he's a grad student in the HUD lab. So I showed you glycolic acid alanine, but Martin synthesized other molecules, glycolic acid glycine, lactic acid alanine, lactic acid glycine. Lactic acid is the hydroxy acid analog of alanine, so we just have the methyl group here as opposed to the glycolic acid. And what we wanted to see is what happens if we are going to make a complicated mixture, but first we just had to make sure that all of these peptides polymerize when we dry them. So you see like glycolic acid alanine, you see the different polymers. Perhaps I didn't mention 2 means 2 units of glycolic acid alanine and so on. So all of these peptides used are forming polymers. And when we looked at the degradation rate of these peptides or Depsipeptides, we note that the ones with the lactic acid, so with the methyl here, are much more stable. So we said, let's take the one with the lactic acid, one with glycolic acid, put them together, and then see if we have selection once we have degradation in the wet phase towards the lactic acid-containing ones. So we took glycolic acid glycine, lactic acid alanine, we dry them, so we form polymers, and then we hydrolyze. After some drying time, we got polymers. So you can see in red polymers of glycolic acid glycine, so that's 2, 3, 4 of glycolic acid glycine. Some homopolymers of lactic acid alanine in green, right here. And then we also got copolymers composed of both glycolic acid glycine and lactic acid alanine, like these ones. Now, once we hydrolyze these Depsipeptides that are formed, we see that after hydrolysis or after degradation time in water, we see that the ones that survive the most are the ones that are enriched with lactic acid. So we were able to show some selection based on chemical stability of Depsipeptides in our system. And I hope I convinced you today that Depsipeptide can be a plausible peptide backbone. First, we're able to polymerize Depsipeptides very readily. We can even see some structures formed by them. And last, really importantly, we're also able to recycle and really sample this huge sequence structural space. So we'll be able to hopefully get some sequences that will give us rise to structures that might be functional. And I don't have time for moving forward, but I'd love to talk to you if you have any questions. Please go to see Martin's poster today, because Martin and I have a library of peptides. These are peptides synthesized by Martin, and we explore some interesting features on these, making the system more complicated, looking for some selection. So with that, I would like to thank the Piazzi. I work with Dr. Hudd, Dr. Williams, and Dr. Grover. I would like to thank my lab members, especially Martin, who have mentioned, the funding agencies, and thank you for listening.
Questions? So I was wondering about the sort of the additional and the reversibility, and how the reversibility is really important, but sort of the enrichment process, and just kind of how you want reversibility, but you also want some sort of progress, if it were. And so what's sort of the balance between those two processes?
Yes, so we really need, the reversibility part is really important for trying out different sequences, but the premise is that if we have some sequences that can fold, they can form some stable structures, and then they will be stable, even though it could be a reversible polymerization. We think that if we have some structures, or if we have binding to a cofactor, for instance, so we have some ways to stabilize the structure ones. So the ones that are not functional, not structured, they will be degraded, put back, recycled into the prebiotic soup, and then we can build some more and to enrich for some Depsipeptides that are more favorable.
Okay, great, thanks. Other questions? Yeah.
So sort of piggybacking on that idea of stabilizing the structure, have you thought about, well, I'm sure you've thought about, but do you have any plans to do things with like different like clays or sediment or other cofactors present, those kinds of experiments?
Yes, this is definitely a direction that we take. We want to be able to select based on structure or function or a binding, for instance. So, yes.
Slightly silly question. Why starting specifically from the dimer starting points rather than just like a mixture of the monomers? Is that just to sort of get things going faster or make the products larger or something? Or
yes. So we kind of know that we had a lot of time for chemical evolution, but we kind of want to speed up the process and see and be able to speed up things. So we think that because we know the chemistry, so we know that we can make these peptides with hydroxy and amino acids, we know that they will be enriched after a wet cycle, after some heating time. We want to kind of just make it more simple, so we'll be able to see things and not working with, at this point, with a complicated mixture that will be they're just harder to analyze, and so gain some insights from them. But we just want to speed up the things to simplify.
Okay, great. Let's thank Moran again.